Welcome to Let's Get to the Marks, going through an A-level biology question. This one's on nucleic acids or nucleotides. Let's get into it. It's relevant to all exam boards, so don't be a little wuss and think, oh, it's not in my exam board. Yeah, well, how do you think they come up with those weird questions you've never seen before in your own exam board? They look at other exam boards. So you want to get the A star. Let's get into it. Nice and easy question, this one. Which labeled structure would contain the element phosphorus in this nucleotide? So which of these parts contains the element phosphorus, elemental symbol P? Is it going to be the circle? Is it going to be the pentagon? Is it going to be the rectangle? Well, hopefully you know all of these off by heart by now. The circle is the phosphate group. The pentagon is the pentose sugar, which its specific name depends on which nucleotide we're dealing with. And then the rectangle is called a nitrogenous base. And you know they come in 5 flavors. You got your 4 for DNA, you got your adenine, thymine, guanine, cytosine, and then of course if you're RNA you have uracil instead of thymine. So that's just running through all of those. So which one of these contains the phosphorus? The correct answer here is the first letter, which is Q. And then we got some bonds. We got a phosphodiester bond and we just got a straight covalent bond here. So let's not worry about all of that. Let's zing down the answer there, Q. That'll get you 1 mark, and let's move on. Nice. You should know all of this stuff off by heart. If you don't know this yet and you're doing AES, make a revision card, draw that with the labels, and then on the back, put the correct answers for each of the letters. Okay, part 2. Structure R is different in a DNA nucleotide compared with an RNA nucleotide. Name structure R in a DNA nucleotide and in an mRNA nucleotide. So let's go up and take a little zing and look at structure R. Right, it's got 5 points, it's a pentagon, it's a pentose sugar. That's the similarity. However, in DNA, it's called deoxyribose sugar, and it has 1 less oxygen atom than when it's an mRNA nucleotide. The mRNA nucleotide does have oxygen in it, but it's got less. So it's known as a ribose sugar and not deoxyribose. So that will get you the mark. They're both pentose sugars. Anything with OSE at the end, lactose, glucose, fructose, they're sugars. Okay, which of the following bases would be found in a DNA nucleotide but not in RNA? I kind of went through that already. I mentioned it. Thymine is not found in RNA. You'd find thymine in DNA, but you'd find uracil in mRNA or tRNA. You wouldn't find thymine or rRNA. The diagram shows a polypeptide chain. We've got these little circles here which represent monomers, small molecules that join together to make a polymer. Number 2 says, first question, which row shows the correct type of monomer and the type of reaction that joins the monomers together? So what is the name of these monomers? Well, they're in a polypeptide. What's a polypeptide? Hopefully you remember it's a protein. And so they don't contain fatty acids. Let's get rid of those too. So the correct monomer is amino acid. How do we join 2 of these together? They're joined by a peptide bond in a reaction called a condensation reaction. All biological molecules are formed in condensation reaction. All biological polymers and large molecules, they're joined together in condensation reactions. And hydrolysis means splitting or breaking. That's the breaking reaction. Which of the following types of bonds are involved in forming the tertiary structure of proteins? So we've got disulfide bridge, ester bond, ionic bond. Well, ester bonds are found in fats, so it's not that. We're talking about proteins here. Maybe that one, disulfide, hydrogen ionic. Disulfide, we've got an ester bond again, that's in fats. And we've got glycosidic bond, that's found in carbohydrates, not in proteins. So we can get rid of that. So our correct answer is B. And yeah, you've got disulfide bonds, hydrogen bonds, and ionic bonds. Essentially covalent bonds, hydrogen bonds, ionic bonds. They form the 3D or tertiary structure of a protein. You can say hydrophilic and hydrophobic interactions as well, which also help form that tertiary structure of the protein, which is extremely important for its function. Right, last part to this question. Describe the role of RNA in the formation of this polypeptide. Essentially, guys, they're talking about how is mRNA and tRNA involved. Well, that's in translation, happens later, outside the nucleus. Transcription happens first, translation happens later. They're asking you to describe translation here, guys. So see if you can put 4 bullet points on translation. Try to put 5 or 6 because we want to be specific as to the role of RNA in translation. Try to keep it focused on that. Pause the video, have a go, make it active. All right, unpause. I'm going to go through the answers. So I don't know why I say unpause, like you can hear me when you pause. Right, anyway, mRNA leaves nucleus via a nuclear pore. That's often the end of transcription, but we might start our bit about translation with that. Even if they say ignore, what does it matter? They're not taking marks off. So mRNA, what does it do then? It joins to the ribosome, right? Attaches to a ribosome. So that's always going to get you on a mark because we're talking about the focus of mRNA and tRNA. Then tRNA brings a specific, we need that word, amino acid dependent on the mRNA codon. And we can also say that the tRNA joins via its codon to the via its anticodon to the codon on mRNA. So the anticodon is on the tRNA and the codon is mRNA, and they complementary base pair. That's definitely a mark, probably 2 marks. Something else which I haven't mentioned here, which would have got us a mark, is maybe potentially would have got us a mark. tRNA has a specific binding site that binds to the amino acid. So I didn't mention the binding site. You don't find that on mRNA. It's one of the differences between the two. tRNA has a binding site. Now, peptide bonds form between the amino acids as they're lined up by the tRNA. As they're brought down by the different tRNA molecules, the different amino acids line up and peptide bonds form. But is it the RNA that makes the peptide bonds? It's not. The RNA doesn't really have anything to do with the peptide bonds. So I would say that is an irrelevant point to the question. Would you lose a mark for that? No. So that's why I'm saying here, I would just write everything about translation and you'll pick up all the marks. At worst, they'll ignore this and you won't get a mark because it's not relevant to the role of RNA in translation in the formation of a polypeptide. So anyway, going back to the RNA, tRNA leaves, the first one leaves, I should say, and a second tRNA arrives, bringing a new amino acid. Now, it might be the same if the codon if the codon's the same on the mRNA, and so which tRNA joins is dependent on the codons on the mRNA. So the sequence of codons or the order of the nucleotides on the mRNA, i.e. if it's CGG or GCG, whatever the order is of the nucleotides on the mRNA, that is going to determine our sequence or order of amino acids. And this forms the primary structure of the polypeptide. And of course, so it's the order of the amino acids that forms the order of the nucleotides that forms the sequence of amino acids in the primary structure of a protein. And then due to the different positions of the R groups, we're going to get different folding, different hydrogen bonding, giving us beta pleated or alpha coil. And then dependent on the R groups, we're going to get disulfide bridges and ionic interactions and things, and we're going to get that specific 3D structure. Anywho, that will get you all the marks. That's the end of the question. Don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.